Welcome once more to my YouTube channel. This is Medical Academy. I hope you guys have been doing well. We are still dealing with our series on hypertension and antihypertensive. This is our fourth episode, and I believe you guys by now you have an idea of what we have discussed when it comes to antihypertensive. In our first episode, we gave an introduction of the meaning and classification and grading of hypertension. In our second episode, we looked on the drugs that work against the sympathetic drive. Remember, sympathetic drive tends to elevate our blood pressure. So, on our first episode, we looked on the drugs that tend to work against the sympathetic drive called sympatholytics and also sympathetic antagonist. So after our second episode, we went on our third episode and looked on drugs that work against the RAS system. Remember, we have a system called the renin angiotensin aldosterone system, which tends to also elevate our blood pressure. So the drugs that go against that system will help us in managing our BP. So today we are going to move forward in our fourth episode. This is going to be slightly larger episode because diuretics is a topic on its own, very large topic, but I decided to include it in our series because some of the drugs that are used as diuretics also are used as antihypertensive drugs. So I decided to include diuretics inside our series, but remember, you can also study diuretics as a whole, as a topic on its own. So today we are going to look at diuretics. It's going to be a long series. So I want you guys to be in a quiet place, have your pens, you have your pen and your notebook as we dig deep on how these drugs work. But before that, we are going to have a small session to remind ourselves on the anatomy and physiology of the renal system. So once we have that understanding, so it will be very easy for us now to just look on the drugs that work on that system. So before we begin looking at the drugs, we are going first to have an intro on the anatomy and physiology of the renal system. So I hope you guys are ready for this session. So before we begin, as I said before, I know you guys have done anatomy and physiology in your first block or in your first year of nursing school or medical school, and I believe you guys have an understanding on the renal anatomy and physiology. But I just want us to remind ourselves on what it entails, so that when we begin looking at the drugs, it will be easy for us to understand the mechanism of action of these drugs. Remember, this is Medical Academy. Please support our channel by liking the video, by sharing the link, and also inviting other friends to watch our videos. This is a session for us to learn, to remind ourselves what to do in class, and also to build our confidence and knowledge when we go for exams. So let's begin. So first of all, I'm going to take us through the review of the renal anatomy and physiology to remind ourselves on what we did on our anatomy class. So as you can see in my board, there's an image that shows the gross and microscopic structure of the human kidney. On the left side, you can see the gross structure. On the right side, you can see we have the microscopic structure or the nephron. So to begin with, on the gross structure of the kidney, this is a cross-sectional of the kidney that you can see. We have three distinct regions that I want you guys to know. We always have the outer region called the renal cortex. We have the renal medulla and the renal pelvis. So it's important for you guys to understand what the role of these sections and what structures or what part of the nephrons are found on each of these sections. So we have our renal cortex, the outer section. This tends to have the renal corpuscles. I'm going to tell you what is the renal corpuscles in our letters on the slides that will follow. And after the cortex, we've got the medulla. The medulla is very special region. You know that it has got this shapes that looks like pyramid. We call them the renal pyramid. It may vary in number. Some may have 7, some may have 8, some may have up to 12, depending on the genetics of the human body. But what you have to know is that this renal medulla also they contain a very crucial section of the nephron. As you can see, this section of the nephron is always found in the medulla, whereas the renal corpuscles, this section here, is always found on the renal cortex. So this our renal medulla, it drains into the renal pelvis. But then again, we have got these structures that are found on the apex of the renal pyramid, which we call them the minor calyx. The minor calyx drains into the major calyx, and they merge to form the renal pelvis, which now drains the urine in the ureter. The ureter will also drain the urine into the bladder into the urethra, and that's how the urine flows. Now, back to our session. Apart from knowing the major sections of the growth structure, we also have to know the vasculature of the kidney. The kidney is always supplied by renal artery and drained by the renal vein. So that being mentioned, then we can look on the microscopic structure. As you can see here, the nephron, actually one kidney contains very many nephrons. That's why a human being can easily survive with only one kidney. So that being said, you can see in this diagram here, the first section that you have is the renal corpuscles, and this is always composed of two major parts. That is the glomerulus and the cup-like structure called the Bowman's capsule. So the glomerulus and the Bowman's capsule, they make up the renal corpuscles. And the glomerulus is majorly formed by two major arterioles, that is the afferent and the efferent arteriole. Yeah, so the thickness of these arterioles tend to propagate the filtration process that happens in the glomerulus. So that's another topic for another day. That being mentioned, once the filtration happens in the glomerulus, the filtrate gets drained into the proximal convoluted tubule. So here also we have got some reabsorption that occurs. I'm going to explain later on the other slides that will follow this slide. So this is our PCT. Our PCT then drains into the descending loop of Henle, into the ascending. So the ascending loop has got two major sections. It has got the thin ascending and the thick ascending loop of Henle. I'm going to explain what happens in those two sections. Apart from that, we also have our last section called the distal convoluted tubule. Also, this has got two sections. It has got the early DCT and late DCT, and finally it drains into the collecting duct as urine. So a lot of process happens in the nephron. I'm going to explain into details what happens in the nephron. So everything that happens leads to urine formation, but that I'm going to explain now what happens in each section of the nephron, because remember, we are targeting diuretics, and diuretics, these drugs, they have different mechanism of action. Some will work in the PCT, some will work in the descending loop of Henle, some will work in the early and late DCT, some will work in the thick ascending limb. So we have to know what happens in each section. So, and once we understand what happens in each section, it's going to be easy for us to understand the mechanism of action of these drugs. So let's move on to our next slide. Now let's begin with the first section called the renal corpuscles. So remember, I told you guys the first section of the renal corpuscles, we have the glomerulus. As you can see in my diagram here, if you extract one nephron under a microscope, you can see the microscopic structure, the nephron, and further you can see this section here. Now, this here is our renal corpuscles, as you can see when magnified. So this is majorly composed of two main sections. That is our glomerulus and also the Bowman's capsule. So remember, our glomerulus is made up of two sections. That is, we put the afferent arteriole, the efferent arteriole, and the glomerular capillaries. And there is where we have our filtration process. So glomerulus is a tuft of capillaries in Bowman's capsule that filters blood and obviously the final formation or the final product will be formation of urine. Its selective barrier keeps proteins cells in blood while allowing small solutes to pass. Remember, we have got the podocytes, and the filtration process tends to happen. We know that the filtrate will be composed of very fine, tiny molecules. And that being said, the larger molecules, the likes of proteins and the RBC, won't be able to be filtered because they're too large to be filtered. Uh, that being said, we also have our Bowman's capsule, the cup-like structure. So it's a cup-like structure around the glomerulus, and with an outer parietal layer and an inner visceral layer, and the inner visceral layer has got some podocytes. As you can see, some swollen, you can see these swollen areas here that you can see. So that's the visceral layer composed of the podocytes and the outer parietal layer, which tends to be thinner, very thin. I can say its epithelium is slightly, maybe the simple squamous, very thin epithelium, so that the filtration process can be much easier. So it collects the first filtrate, and once the filtrate is collected, it's directed into the proximal convoluted tubule. I hope we have an understanding till there. So in this slide, our take on number one is the understanding of the renal corpuscles. Number one, we know that this is always found in the renal cortex. Even in this diagram, you can see we have extracted this section from the renal cortex. So this section here is the renal cortex, and down here we have the renal medulla. So the renal corpuscles is always obstructed from the renal cortex. Number two, we know that it's majorly composed of two main parts, and that's our number one, glomerulus. Number two, the Bowman's capsule. I hope that message is taken home. And one thing that I have to know, the major activity that happens here is the filtration process. Now, our second section now is the proximal convoluted tubule. So I want us to be very patient in this slide because I'm going to explain the physiology mechanism that tends to happen when we mention the PCT. So in the PCT, we always have reabsorption of various molecules. Just to begin with, we know that glucose is always completely reabsorbed in the PCT, and that being said, it depends on the physiology or the hemostasis. When a client is hyperglycemic, we may see the body allowing glucose to flow into the urine. That's why those clients with diabetes, they may have glucose in urine, also called glycosuria. Okay, that being mentioned, you have to understand the sections of the PCT. So this table diagram shows the three major sections. Now, here we have our tubular lumen. Yeah, tubular lumen. Here is where we have the filtrate. This one here is the PCT epithelial cell. Yeah, this is the PCT epithelial cell, and here we have our interstitium and the capillary, or a section that leads into the blood. So this section leads into the blood. This is our cell, and this is the filtrate. So we are going to explain how some of the filtrate, or some of the molecules that are found in the filtrate, how those molecules in the filtrate move from the filtrate into the blood cavity. So I'm going to explain some of the important molecules that you have to understand. And this being said, there's some drugs that act on some of the enzymes, or they lead to variation of various molecules. So that being said, I'm going to explain what happens here. So let me just highlight a marker pen. Let me use a red pen. Yeah. Now, I want you guys to focus on two major areas. I want you guys to focus on bicarbonate reabsorption. I want you guys to focus also on sodium reabsorption, and also hydrogen excretion. So those three major areas. Now, let's begin. What happens? We have this exchanger here. We call it sodium hydrogen exchanger. So this, what it does, it brings sodium inside and brings hydrogen ion outside. So this exchanger, what it does, it brings sodium inside the epithelial cell, inside the PCT epithelial cell, and takes hydrogen outside. Now, this is very important. Why? Because the hydrogen that is released will really help in the reabsorption of bicarbonate and also sodium, as you have seen in the diagram, in my diagram below. Now, when the hydrogen is released, it combines with the bicarbonate. So this hydrogen combines with the bicarbonate to form, this is called carbonic acid. Back to our chemistry class that you did in high school. If you take bicarbonate, which is an anion, and hydrogen ion, which is a cation, combined together, we will form a carbonic acid. I hope you guys are together. So as you can see, my first point here, filtered bicarbonate in the tubular lumen combines with a secreted hydrogen ion to form a carbonic acid. I believe we are together. So that's the first process. The hydrogen that has been released combines with the bicarbonate in the filtrate to form carbonic acid. That's our first step. Now, let's move in the second step. We have an enzyme called carbonic anhydrase. To be more specific, carbonic anhydrase 4, because we have got two types of carbonic anhydrase in this process. We have got number one, carbonic anhydrase 4, which is found on the tubular lumen or in the filtrate. And we also have carbonic anhydrase 2, which is found intracellularly inside the epithelial cell. So that's a point that you have to note. Now, our carbonic anhydrase 4, what it does is that it dissociates the carbonic acid to form carbon for oxide and water. So luminol carbonic anhydrase or carbonic anhydrase 4 rapidly converts carbonic acid to CO2 and H2O. Now, our CO2 can easily penetrate the cell, so it will enter the cell. Our CO2 will enter the cell, and also water can also enter the cell. So these two will easily enter the cell. So the CO2 and water now will enter the cell. Now, what happens? Now, inside the cell, the CO2 and water will now combine under the influence of carbonic anhydrase 2 to form carbonic acid. So we have formed carbonic acid inside the cell. Amazing. We have formed carbonic acid inside the cell, and this began here, whereby we released hydrogen ion. The hydrogen ion merged with bicarbonate to form carbonic acid. The carbonic acid dissociated to CO2 and water. The CO2 and water went or passed into the cell, and they merged in the cell to form carbonic acid under the influence of carbonic anhydrase 2, which is found intracellularly. Now, once we have our carbonic acid, this will again dissociate to form hydrogen ion and bicarbonate ion. So our cation, the hydrogen ion, will now be eliminated via this pump outside the cell, as you can see here, in the exchange of sodium ion. So this hydrogen ion that has been formed here now will propagate or will promote the function of this exchanger, whereby it will be excreted outside in exchange of sodium ion. The bicarb now can easily be reabsorbed in the blood. It can easily be reabsorbed in the blood. Now back, we always have our sodium ion that is reabsorbed here. So this sodium ion that is reabsorbed, what happens? It's always moved into the blood via the sodium potassium ATPase pump. So the sodium potassium ATPase pump, what it does is that it removes 3 sodium ions outside to the blood in exchange of 2 potassium ion. So the sodium potassium ATPase pump will promote the reabsorption of sodium into the blood by exchanging, by taking 3 sodium to the blood and 2 potassium ion in the epithelial cell. So that's how we have the sodium reabsorption and the bicarbonate reabsorption. Why is this important? This is important because we have got a drug. This is a drug that tends to work against carbonic anhydrase enzyme. there's a carbonic anhydrase inhibitor drug. So a carbonic anhydrase inhibitor, this drug, for instance, one of the drugs is called acetazolamide. So this drug, what it does, it tends to inhibit this process here. It comes and stops this process. What happens? This process is inhibited, and also this process is inhibited by carbonic anhydrase inhibitor. So one thing that you have to note is that for sodium reabsorption to happen, we need hydrogen ion. Yeah, and also for backup ion to be absorbed, we need hydrogen ion. So if you block this process, if this process is stopped, it means carbonic acid will be released in urine, or bicarbonate ion will be released in urine. There will be a bicarbonate release in urine. So if you stop carbonic anhydrase enzyme from functioning, bicarbonate will be released in urine. And we know from our physiology, if you promote bicarb excretion, you basically will lower our body pH. You will promote metabolic acidosis. And that being said, also, you will make the urine to be more alkaline, and that will further propagate the formation of renal stones. Yeah. So carbonic anhydrase enzyme number one, they promote the excretion of bicarbonate ion. Yeah, and same otherwise they also promote the excretion of hydrogen ions and also the excretion of sodium ions, and that will lead also to uh, uh, excretion of water and sodium ions. Yeah, so that's something that you have to note. That's a very important point. So if we stop our carbonic anhydrase from functioning, it means that our bicarb ion will be released in urine. They won't be reabsorbed, because remember, for it to be reabsorbed, we need carbonic anhydrase. Our carbonic anhydrase needs to propagate the formation of carbonic acid in the cell, which will dissociate into hydrogen ions. The hydrogen ions will propagate the reabsorption of sodium and also the reabsorption of bicarbonate. So that's how the whole process happens. So I believe you guys have understood. So let's move to our next slide now to explain it further. Now in the PCT, as I've explained, you have to know that the reabsorption of bicarb is very essential. I have explained how this happens, as that hydrogen ion is released in urine. Let me just point my highlighter back. So remember what I've mentioned in my previous slide, as that hydrogen ion is this exchanger which brings hydrogen outside in exchange of sodium. So once you release hydrogen ions outside, it merges with bicarb to form carbonic acid. The carbonic acid dissociates in the presence of carbonic anhydrase enzyme or carbonic anhydrase enzyme number 4 to form water and CO2. The CO2 will be the CO2, because CO2 is permeable, they will flow into the PCT cell, and once inside here they will merge with water to form carbonic acid. The carbonic acid, because it's unstable, it will form hydrogen ions and bicarb ions. The bicarb ions will be reabsorbed into the blood, whereas the hydrogen ions will be expelled via this exchanger. Remember also, sodium ion can be reabsorbed in the blood via the sodium potassium ATPase pump. So that's what happens in the PCT. So one thing that is very key is that decent amount of sodium reabsorption occurs. Around 65% of sodium reabsorption happens in the PCT. This is very crucial. So if at all there's a diuretic that inhibits sodium reabsorption, it can be very massive in terms of diuresis, because a lot of sodium is reabsorbed in the PCT. So inhibiting sodium reabsorption will lead to massive sodium and water excretion that I've explained. So a diuretic that specifically works here is a diuretic that inhibits this enzyme, the carbonic anhydrase inhibitor. And I've told you guys that that drug is called acetazolamide. I will explain later on how it works and the indications and the contraindications of using a carbonic anhydrase inhibitor. So apart from all these shenanigans that I've mentioned here, you have to remember that glucose and amino acid also are reabsorbed in the PCT and bicarbs, as I have explained. Now let's move to the descending limb. After the PCT, the filtrate moves to the descending limb. Now, in the descending limb, one thing that I have to note is that descending limb tends to be located in the renal medulla. And now, this filtrate is somehow isotonic, but the medulla tends to be much highly concentrated. So, because the medulla is hyperosmotic, what happens? The water, this section here is always permeable to water and impermeable to solid. So what happens here is that water will move via down the osmotic gradient. So there will be an osmotic gradient. And now water will move from a lowly concentrated area in the filtrate to a highly concentrated area in the medulla. And now what happens is the reabsorption of water molecules. That's what happens in the descending limb. So only water is reabsorbed here because this section is permeable to water but not to solid. So we don't have solid reabsorption here because this section is impermeable to solid but permeable to water. So it plays a very key role in the kidneys urine concentrating ability. Remember, as you remove water, you make the filtrate more concentrated. So the urine will be more, the filtrate will be more concentrated as it moves along the descending limb. Yeah, so that's something that you have to note. So that being said, let's move now to ascending limb. And ascending limb, I want us to focus more on the thick ascending limb. Now back to our another cascade or another physiology class. As you can see here, let me just put my marker pen on. Let me put our red marker pen on. Now, in this diagram here, I want you guys to note three crucial sections. We have our tubular lumen, where we have our filtrate. We have our epithelial cell, the thick ascending limb epithelial cell. And here we have our interstitium or medulla, which then will be in touch with our capillary or blood. Now, those are our three major sections. Now, here you have the filtrate here. This is our epithelial cell, and here is our interstitium or our medulla. I hope you guys are together. I hope you're together. Let me just put back our eraser. Our last point. Now, here we have our filtrate or tubular lumen. This is our epithelial cell, or it can be more specific, a thick ascending limb epithelial cell, and here we have our medulla. I hope you're together. Now, there's a very crucial channel that I want you guys to note. A very crucial channel that I want you guys to note. We have this channel here, or a co-transporter. This one here is called sodium potassium chloride co-transporter, NKCC2 co-transporter. And here we have a sodium potassium ATPase pump that I'm going to explain later. Now, let's focus here. What happens? So this co-transporter, it takes sodium, it reabsorbs sodium, as you can see. It reabsorbs potassium and chloride together. So our sodium ion moves, our sodium ion, potassium ion, and chloride ion will be transported from the tubular lumen or from the filtrate inside the cell by this transporter. So here we will have sodium ions that will be moved inside here, sodium ions. We will have our potassium ion, and also our chloride ion that has been moved by our co-transporter. So the sodium ion that has been moved inside will again be taken into the medulla via the sodium potassium ATPase. This pump, you know, it takes the sodium ion inside in exchange of potassium ions. So it will take 3 sodium ions and exchange 2 potassium ions. So 3 sodium ion will be moved into the medulla, and 2 potassium ions will be moved from the medulla inside the epithelial cell. Now, that being said, our chloride ion will be moved into the medulla. Now, there's one thing that you have to know. We have a lot of potassium inside this cell. Why? Because this co-transporter brings potassium inside, and this sodium potassium ATPase pump also brings potassium inside. So there's a potassium ion, this potassium ion brought by the co-transporter, and there's also these two potassium ions that is being brought by the sodium potassium ATPase pump. So we have a lot of potassium inside here. And because you have a lot of potassium, what happens? The potassium will move down its concentration gradient via this channel here. So potassium will move outside because there's a lot of potassium inside the epithelial cell. So when the potassium moves outside, the potassium will make the potassium will increase the positive charge outside here. There will be an increase in the positive charge, and this will be caused by the movement of potassium from the epithelial cell into the filtrate. Now, because there's an increase in the positive charge, what happens? We have our divalent cations, and the divalent cations here is the calcium and the magnesium. So an increase in the positive charge in the lumen will repulse the cations, the divalent cations, the calcium and magnesium, to be reabsorbed in between the tubular, in between the epithelial cells. So there will be movement of calcium and magnesium also into the medulla. So in short, that being said, in the thick ascending limb, we have, in the thick ascending limb, we will have the movement of sodium ion to the medulla, the movement of potassium to the medulla, the movement of chloride to the medulla, and also the movement of calcium and magnesium to the medulla. That's why a diuretic that impacts this channel, like the loop diuretics, they always inhibit this channel. And if you inhibit this channel, it means you will excrete sodium ions, you will excrete potassium ions, you will excrete chloride ions, you will excrete calcium ions, you will excrete magnesium ions. I hope we are together. Very, very interesting. Let's move to our next slide. If you haven't understood, please just go back and forward on what I'm trying to explain. That being said, let's move to our next slide. Now, I've just explained what happens in our ascending limb of loop FN. That's a simpler form of the diagram that I was going to explain. So this co-transporter here is what I was going to explain. This co-transporter, it takes potassium, sodium, and chloride ion inside. This sodium ion will be exchanged via the sodium potassium ATPase pump. So this sodium ion will be taken into the blood via the sodium potassium ATPase, which exchange sodium with potassium. So there will be potassium movement into the cell. Yeah, the chloride ion will just move via their channels. And that being said, there will be a lot of potassium. Number one, there will be an increase of intracellular potassium. And because there's high potassium in the cell, the potassium will move down its concentration gradient via these channels here. And because when that happens, there will be an increase in the positive charge in the lumen. Yeah, and an increase in the positive charge. Remember, positive ion repels positive ions. So the calcium and the magnesium ions, the divalent cations, will move in between the cells to the blood. So that's what I was trying to say. So in short, we have got sodium reabsorption, potassium reabsorption, chloride reabsorption, calcium reabsorption, magnesium reabsorption. So a diuretic that stops this channel will lead to sodium in urine, potassium in urine, calcium in urine, magnesium in urine, chloride in urine, and also final product, water in urine. So there's some sodium reabsorption and decent amount of water reabsorption. About 25% of sodium is reabsorbed in this section of the renal tract. So remember, on our PCT, we had 65, and here you have 25. So far, 65 plus 25, that's around 60, that's 90%. So till here, we have reabsorbed 90% of sodium back to the blood. Just imagine, 90%. So the thick ascending limb of the loop of Henle relies on highly metabolic active transport via the sodium potassium chloride co-transporter to aggressively pump out solute, making it the primary target for loop diuretics. Yeah, so loop diuretics work by inhibiting this co-transporter, and that's why loop diuretics are they're massive when it comes to dealing with edema, or when it comes to excreting massive fluids. Loop diuretics tend to do a very good job, the likes of furosemide. I'm going to explain later on how that happens. So in contrast, the thin ascending limb. So the thin ascending limb, remember in ascending limb we've got the thin and the thick. So the thin ascending limb is not so much active, remember, because what happens? The thin ascending limb relies on passive diffusion, and this is the first section. So remember, the filtrate in the descending limb was concentrated because water was reabsorbed. So when water was reabsorbed, the filtrate became highly concentrated. And so that being said, because of the highly concentration in the thin ascending, what happens? We experience passive diffusion of the sodium and chloride ions. So that here we don't have a huge explanation. I just want you guys to focus more on the thick ascending limb, because in the thick ascending limb is where our loop diuretics work. So the mechanism of action of loop diuretics tends to occur on the thick ascending limb. Yeah, last section, the loop diuretics, they inhibit our sodium, potassium, chloride co-transporter. So let's continue. Now we have got the distal convoluted tubule, as you can see in our diagram below. So we have explained what happens in the renal corpuscles. We have explained what happens in the descending limb. We have explained what happens in the ascending limb, both the thin ascending limb and the thick ascending limb. Right now we want to explain what happens in our distal convoluted tubule. But remember, in our DCT, we have got two sections. We've got the early DCT and the late DCT. So distal convoluted tubule, we have both the late and early distal convoluted tubule. So in both of these sections, there is going to be some degree of sodium and water reabsorption. So the absorption can be inhibited by number one. So early DCT, what happens? In early DCT, we have 5% of sodium reabsorption. So the 90% plus 5, that's 95%. And late also has got 5%. So in short, once the filtrate leaves the DCT, there's 100% of sodium reabsorption that has just occurred. Remember, 65% of sodium reabsorption happens in the PCT, the proximal convoluted tubule. 25% of sodium reabsorption happens in the ascending limb of Henle. 5% and the 10% will happen in the distal convoluted tubule, whereby you have got 5% happening in the early DCT and 5% happening in the late DCT. So we are going to look for this being mentioned. We are going to look on how the sodium reabsorption happens in the early DCT, and also how it happens in the late DCT. So our last point here, in distal convoluted tubule, especially the late DCT, sodium reabsorption may be dependent on either aldosterone hormone or the ADH for water reabsorption. So these are very crucial points that you have to understand. Remember, when we learned our RAS system, I told you guys that in the RAS system, the aldosterone hormone plays a crucial role in sodium reabsorption. So that you have to remember. Remember also our vasopressin, the ADH, it also plays a key role when it comes to water reabsorption. So that's something that you have to note. Now, back to our class. Yes. Now, let's take our marker pen, a red marker pen. Yeah. Now, this diagram here, you can see this is our early distal convoluted tubule, or our early DCT. And this diagram has got three sections. We have got our tubular lumen here. This is our tubular lumen, whereby we have our filtrate. This is our tubular, so this is our early DCT epithelial cell, as you can see here, and this is our peritubular capillary. So we've got three major sections. Now, in this tubular lumen, we have our filtrate. This is an epithelial cell. Yeah, you should know the content of the epithelial cell. and here we have got our interstitium and our blood. Now, there's something that I want you guys to note, and the most important channel or co-transporter that you guys should know here is this one, the sodium chloride co-transporter. Where's my pen? Where's my pen? Can you see my pen? Yeah, here it is. Yeah. So I want you guys to note this co-transporter. This is the most crucial point. This co-transporter. I want you guys also to note this sodium potassium ATPase. This is very crucial. Yeah. Yeah, and also this sodium calcium exchanger. These three. So these are very crucial areas. Now, it begins here. What happens? We have a co-transporter here called sodium chloride co-transporter. This co-transporter, it reabsorbs sodium and chloride ions. So sodium ion and chloride ion will be moved from the filtrate to the epithelial cell by the sodium chloride co-transporter. So we have our sodium inside and our chloride inside. So when the chloride ion move inside, the chloride ion can just move via its channel. The chloride ion will be moved to the blood via its channel. Yeah. On the other hand, the sodium ion can be moved, number one, via the sodium potassium ATPase pump, which will take 3 sodium outside the cell in exchange of 2 potassium inside the cell. So our sodium potassium ATPase pump will further remove sodium ions in exchange of potassium ions. So that being said, we always have our exchanger here. When we have a low sodium concentration inside the cell, this exchanger will bring sodium inside the cell and take calcium from the cell to the blood. So the sodium calcium exchanger, it depends. When there's low sodium in the cell, it will bring more sodium inside. And remember, when it brings sodium inside, it takes calcium with it. So that's what happens. So don't forget, we have got our sodium calcium co-transporter, we have got our sodium calcium exchanger, and also we have got our sodium potassium ATPase pump. Now, there's something that I want you guys to note. There's something that I want you guys to note. There's some diuretics that work on this co-transporter, and this diuretic is called thiazide diuretic. So thiazide diuretic, what it does, it inhibits this co-transporter. Now, what happens if you inhibit this co-transporter? You will limit or you will reduce the amount of sodium being reabsorbed, and you will promote sodium excretion or sodium in urine. So when we have less sodium, less sodium inside here, this will propagate this sodium calcium exchanger. When there's low sodium here, this exchanger will further bring more sodium inside and further promoting more calcium to the blood. So there will be calcium retention. So if you stop this channel here, you will be promoting calcium exchanger. There will be a lot of calcium being driven from the cell to the blood in exchange of sodium ions. So that being said, our thiazide diuretic, they always promote sodium and chloride excretion, but by doing that, they also promote calcium retention. That being said, we also have a very crucial channel, this calcium channel here. This channel, it's always activated by, number one, our parathyroid hormone, or our low calcium or low phosphate. So when we have got an increase in parathyroid hormone or low calcium and low phosphate, this channel will be activated and it will promote calcium reabsorption. So remember what happens in this section. Number one, sodium reabsorption and calcium reabsorption, sodium reabsorption and chloride reabsorption propagated by the sodium chloride co-transporter. We also have got the sodium potassium ATPase pump, which promotes the reabsorption of sodium to the blood. We also have the sodium calcium exchanger, which promotes calcium retention in the blood. So remember, if you stop this channel, you will propagate calcium retention. That's why thiazide diuretics, they always promote calcium retention. So that's something that you have to note. So early DCT, I've explained what happens in early DCT. This is a simple diagram. There's this exchanger. sodium chloride exchanger, which brings in sodium and chloride inside the cell. When they are inside the cell, the sodium ATPase pump will further take sodium into the blood in exchange of potassium ion. And when we have low sodium concentration, our calcium sodium exchanger will be active. And what it does is that it brings sodium inside in exchange of calcium to the blood. So that's something that we have to know or to note. So there's a special transporter in the cell membrane called the sodium chloride cotransporter, as you can see in my diagram here. It takes sodium and chloride into the cell that I've explained. So the chloride ion will move into the blood via the chloride cotransporter. In my previous slide, you have seen how that happens. So the sodium ion moves into the bloodstream via the sodium potassium ATPase. This I have explained. I've said that when the sodium moves into the tubular cell, the sodium ATPase pump will further move it into the blood via the in exchange of potassium. That I've explained. When moving sodium and chloride into the blood, this will also move water. That happens further when you go down the renal tubule. That happens. So the calcium reabsorption is dependent on parathyroid hormone. Yes, parathyroid hormone also may propagate calcium reabsorption. It's a channel that is activated when there's a elevation of parathyroid hormone. Remember, in our endocrine class, we taught you guys that PTH, parathyroid hormone, increases calcium in urine, whereas calcitonin hormone decreases calcium in urine. So that's something that you have to note, or I believe you guys were taught in your endocrine class when you did endocrine physiology. So when calcium moves into the cell, it moves to the blood via a transporter that pumps calcium into the blood and sodium into the cell down its concentration gradient that was caused by sodium potassium ATPase pump. Yes, this point I've explained. This is the pump here, the pump that takes calcium into the blood in exchange. This is the pump here. The pump that takes calcium into the blood and removes in exchange of sodium ions. So I've explained this slide in our previous slide. So if you haven't understood, please just forward. This is a monologue. You can just forward back and replay what I've just explained. I hope you're together till there. So I've explained this pump, the sodium calcium exchange pump, and this pump is what leads to calcium retention. Yeah. Thiazide diuretics inhibits the calcium chloride cotransporter. So the diuretic that inhibits this cotransporter is a thiazide diuretic. So thiazide diuretic will inhibit this cotransporter or this exchange. Now, on our next slide, now I want to explain what happens in the late DCT. So I've explained what happened. I've explained what happens in our early DCT, whereby our thiazide diuretics work. And now I want to explain what happens in our late DCT. Now, what happens in our late distal convoluted tubule? There's three things that I want you guys to note, or three channels. The first one is ENaC channel. ENaC means epithelial sodium channel. I want you guys also to note the sodium potassium ATPase pump. And lastly, the potassium channel. So you have got the sodium channel, the ENaC. We have the sodium potassium ATPase pump, and we have our potassium channel down here. This diagram, we have got three sections. You have got a tubular lumen. Here we have our filtrate. This is our principal cell, and here we have our interstitium, or maybe our peritubular fluid or our blood. This is our blood section. Now let me take my marker pen. Let me use my marker pen. Now we will focus on the ENaC. We will focus on the sodium potassium ATPase, and we'll focus on this potassium channel. Now what happens? Now we always have a hormone called aldosterone hormone. Remember in our previous series, on our previous episode, I taught you guys what happens in the RAS system. There's a hormone called aldosterone hormone. This hormone always propagates sodium reabsorption in the late DCT. When the aldosterone hormone is released, the aldosterone hormone always propagates the formation of three channels. The aldosterone hormone always propagates the formation of ENaC channel or epithelial sodium channel. it also propagates the formation of, let me just highlight. So aldosterone hormone leads to formation of 3 channels. Number 1, that's the NAC channel, the sodium potassium ATPase, and also the potassium channel. Now what happens? Now this happens in the principal cell. We know the sodium channel will promote the reabsorption of sodium ion. So sodium ion will be reabsorbed in the principal cell via the NAC channel or the epithelial sodium channel. When sodium has been reabsorbed, the sodium will be reabsorbed further to the blood by the sodium potassium ATPase pump. The sodium will be taken, the 3 sodium ions will be taken to the blood or into the interstitium or the peritubular fluid in exchange of 2 potassium ions. So the sodium that has been reabsorbed will be further reabsorbed via the sodium potassium ATPase pump in exchange of the calcium ion. So there will be an increase of potassium. There will be an increase of potassium ion in the principal cell because this has been promoted by the sodium potassium ATPase pump. Because remember, when the sodium potassium ATPase pump is active, it takes 3 sodium to the blood and 2 potassium in the cell. There's an intracellular exchange whereby potassium ion will be moved into the cell. So this cell will have an increase of potassium ion. And when there's an increase of potassium ion, the potassium ion will move down its concentration gradient via the potassium channels. So that being said, the aldosterone hormone promotes sodium reabsorption to the blood and it promotes potassium excretion. So aldosterone hormone promotes sodium reabsorption and potassium excretion. I repeat again, aldosterone hormone promotes sodium reabsorption and potassium excretion. And how does this happen? I've explained. Is aldosterone hormone promotes formation of NAC channel, sodium potassium ATPase, and also potassium channel. And when sodium is reabsorbed, the absorbed sodium will be exchanged with potassium. There will be an increase of potassium in the principal cell, and increased potassium will promote diffusion down its concentration gradient via the potassium channel. So that's what I've explained. And same happens in the late DCT, and it can also happen in the collecting duct. So this is the same happens into the early stage of the collecting duct. So these are the image of collecting ducts that same process tends to happen. Yeah, whereby the sodium moves inside by the NAC channel, and when the sodium is inside, what happens? The sodium potassium ATPase pump will take sodium to the blood in exchange of potassium ions. The potassium ions will lead to an increase of potassium concentration in the principal cell. So the increased potassium concentration will lead to movement of potassium from the principal cell to the blood. And that's why we always say our aldosterone hormone promotes sodium reabsorption and potassium excretion. That's what I've just tried to explain. Yeah, as you can see here, NAC promotes potassium entry. Sodium ATPase promotes 3 sodium to the blood out and 2 potassium in, and calcium channel will promote potassium secretion to the blood, to the urine. So potassium will be secreted into the urine. Yeah, there will be a lot of potassium in urine. So that being said, aldosterone antagonist. Aldosterone antagonist, those drugs that work against aldosterone hormone. When there's aldosterone antagonist, there will be no aldosterone produced. And when there's no aldosterone produced, the NAC channels will not be formed. The sodium potassium ATPase pump won't be activated, and the potassium channel won't be much active. So what happens when there's a aldosterone hormone antagonist? No sodium will be absorbed. No sodium will be reabsorbed. And when there's no sodium reabsorbed, the sodium potassium ATPase pump won't be much active, and that means that this channel won't be active. And when this channel is not active, potassium will be retained. So there will be retention of potassium. So by going against the aldosterone hormone, it means you will promote potassium retention, you will promote sodium excretion. will promote sodium in urine. So that's a point that you have to remember. Let's continue to our next slide. So our lead DCT, I've explained what happens. So all this I've explained. So this is our DCT. It may happen in our late DCT and also on the early stages of the collecting duct. So that's something that you have to remember. So I've said that movement happens from the lamen principal cell to the blood. That's just a highlighter. So principal cell is much dependent. So the principal cell is dependent on the hormone aldosterone. So aldosterone moves from the blood into the principal cell, promoting the synthesis of 3 protein channels. That is the ENaC channel, the epithelial sodium channel, the ENaC channels, the sodium potassium ATPase, and the potassium channel on the apical cell membrane. So when that happens, what happens? So the ENaC channels will reabsorb sodium into the cell, and when sodium is absorbed, the sodium will later be pumped into the blood where the sodium potassium ATPase pump. So the sodium will move from the urine, from the filtrate to the principal cell via the ENaC, and when it's inside the principal cell, the sodium will move from the principal cell to the blood via the sodium potassium ATPase pump. So the increased potassium in the cell, which we know has been propagated by the sodium potassium ATPase pump, will move down its concentration gradient into the lumen via the calcium channel. So the sodium reabsorbed, reabsorbed in presence of ADH will also lead to water reabsorption. So we know ADH, the vasopressin hormone, tends to promote water reabsorption also. So this process will lead to less potassium into the blood and more into the urine. So remember, aldosterone leads to less potassium in blood and more potassium in urine. In short, hormone aldosterone promotes potassium in urine. There will be a lot of potassium in urine. Yeah, that's something I have to note. So potassium. So potassium sparing diuretics are the drugs that have a mechanism of action on the late DCT. So potassium sparing diuretics will act on the late DCT by preventing, number one, either by going against aldosterone or via ENaC blockers. So some may work on ENaC blockers. And remember, when the drug act on the ENaC blockers, they will prevent the reabsorption of sodium ions. So that's something that you have to note. So that's done. Now let's look on the drugs. I've explained the physiology of the renal system. Now let us look on the drugs that work on the renal system, the diuretics. Let's begin. So you can, so for now, I think it's almost 1 hour. If you are with me, you can stretch, maybe take a fresh air outside as we refresh, because that was a very long topic. So you can stretch wherever you are. Stretch your hands, breathe in, breathe out, take a slight break, take maybe a short break, stretch as we now look on the drugs. I've stretched enough. Now let's look on diuretics. Now, what is diuretics? So diuretics are a class of medications designed to increase net urine output by promoting natriuresis. This means the excretion of sodium, and diuresis, the excretion of water. So the major mechanism of action of diuretics is to promote sodium and water in urine. It releases more sodium and water in urine. So that's one of the major mechanism of action. So they primarily work by blocking specific ion transporters or channels along the nephron of the kidney. I believe you guys, for those guys who are active during my previous slides, I have explained the major sites under which these diuretics act on. I began with the PCT. We looked on the descending limb. We have looked on the ascending limb, both the thin ascending limb, the thick ascending limb. We have looked on the early DCT and the late DCT and the collecting duct. So I believe, guys, for those who were alert during my previous slides, we have explained some of the transporters that are involved in the entire process. So when you disrupt the reabsorption of electrolytes when you disrupt the absorption of sodium and water, what happens? You will lower the intravascular volume. So diuretics lead. So the major aim of diuretics, the reason why they're also used as antihypertensive, is because they always lead to lower intravascular volume. When this lower intravascular volume, there will be a decreased venous return. A decreased venous return will decrease our preload. So there will be a decreased cardiac preload. And when our preload is and when there's a decreased cardiac preload, what happens? A decreased cardiac preload will a decreased cardiac preload will decrease our stroke volume. And when you decrease our stroke volume, you will decrease our cardiac output and decrease our blood pressure. So that's how these drugs act. The major aim is to lower the intravascular fluid volume. And when you decrease the blood volume, you will decrease the cardiac preload, you will decrease the cardiac output, and you will decrease the blood pressure. So that's why these drugs are always used uh, as antihypertensive. So we have got 5 primary classes of diuretics that you guys should know based on the specific sites of action along the nephron. So the first one that we have is the carbonic anhydrase inhibitor. For those guys who were active in our previous first slides, we looked on how the bicarbonate ion is reabsorbed in the PCT, and when we mentioned the entire process, I mentioned the enzyme involved, the carbonic anhydrase enzyme, and I told you guys when you bring in an inhibitor, a drug that inhibits that enzyme, you will promote bicarbonate excretion in urine. So remember, and when you promote bicarbonate excretion in urine, you will promote metabolic acidosis. So that's something that you have to remember, and I mentioned it in our previous first slides. We also have osmotic diuretics. This majorly act on the descending limb. Remember, in the descending limb, I told you guys that it's majorly permeable to water, so there will be a lot of water reabsorption in the descending limb. And remember also in the descending limb, one thing that you have to remember is that the descending limb is impermeable for solute molecules. So we don't have any solute reabsorption happening in the descending limb. So that's something that you have to remember. We also have got our loop diuretics. This I mentioned when we looked on the physiology that happens in the thick ascending limb. Remember our co-transporter, our sodium, potassium, chloride, all those transporter that I mentioned. So I mentioned that when you block that transporter, there will be a lot of sodium being released in urine, there will be potassium in urine, there will be calcium, magnesium in urine. So I explained how that happens, but we are going just to have a recap on what we have discussed. And also we have got diuretics. I mentioned this when we mentioned our early DCT. When we mentioned what happens in the early DCT, the sodium chloride co-transporter, and how this drug, they inhibit or they block that co-transporter. I will mention this as we go by in our next slides. And last, we have got our potassium sparing diuretics. So amongst the five, the last three are very common. I'm sure you guys have had the loop diuretics being used, the thiazide being used, the potassium. The reason why I'm saying the last three is because when it comes to hypertension or blood pressure, the drugs under these last three classification have been used. The first two, not so much. In osmotic diuretics, we have the mannitol, which is not so much used as antihypertensive. It's used on other indications. We're going to explain on other indications that are being used. But I want you guys to remember one thing is that when discussing diuretics, the last three classifications, the potassium sparing diuretics, the thiazide diuretics, and some of the loop diuretics may be used alongside antihypertensive drugs. But the first two, the carbonic anhydrase inhibitor and the osmotic diuretics, not so much. So that's something that you have to note as we go by with our class. So let's begin with the first one, the carbonic anhydrase. So remember, in this episode, the reason why it's going to be the largest episode is because I have incorporated diuretics. Yeah. So despite learning antihypertensive, one can also learn diuretics in this episode. That's why this episode is so much special compared to the other three episodes. So that being said, let's begin with our first drugs. So the first one, we have carbonic anhydrase inhibitors. So the drug here, the drug that tends to work, or the drug that is known here, is the drug by the name acetazolamide. So that's the drug that you can see. It's this is an oral formulation, 250 milligram acetazolamide. It's not used. Okay, one thing that you have to note. This is a comprehensive episode. So some of these drugs that I'm going to list here, they are not antihypertensive drugs. Yeah, they have got other indications. But because I'm teaching diuretics, I have to bring them in. So that's something that you have to note. Don't get yourself confused. So I said this: the site of action of this drug is this drug acts on the proximal convoluted tubule. This is where it works. This is the site of action. It acts on the proximal convoluted tubule. And what is the mechanism of action? As I said before, this drug targets carbonic anhydrase, both the carbonic anhydrase that is found in the cytoplasmic. So the cytoplasmic carbonic anhydrase enzyme is the carbonic anhydrase enzyme number 2, which is found inside the proximal convoluted epithelial cell. That is the carbonic anhydrase enzyme 2. The luminol one, the luminol carbonic anhydrase, that which is found in the filtrate or in the lumen. This one is the carbonic anhydrase number 4. So if you inhibit this enzyme, it means you will be preventing the bicarb absorption, the bicarbonate reabsorption. So that's something that you have to note. By inhibiting this enzyme, you are inhibiting bicarbonate reabsorption. So that's something that you have to note. So normally, a carbonic anhydrase catalyzes the conversion of carbonic acid into CO2 and H2O in the lumen to allow for bicarbonate reabsorption. This I've explained in my previous slides how this happens. So if you haven't watched that, just forward backwards and listen to what I explained when explaining what happens, when explaining the physiology of proximal convoluted tubule, what happens in the bicarbonate reabsorption. So if you haven't watched that section, please just forward backwards and rewatch. So by blocking the carbonic anhydrase, the cell cannot generate the intracellular hydrogen needed to drive the sodium hydrogen exchanger. Remember, uh, by blocking carbonic anhydrase enzyme, you're not only preventing the bicarbonate reabsorption, you are also promoting the sodium and hydrogen, the sodium reabsorption. How? Remember, I told you guys that the bicarbonate, the carbonic anhydrase enzyme, the one that is found inside the cell, the cytoplasmic carbonic anhydrase, it always lead to the formation of hydrogen ion and bicarbonate ion. So the hydrogen ion that is formed, it is always exchanged by the sodium hydrogen exchanger. The exchanger, it brings hydrogen outside in the filtrate and takes the sodium ion from the filtrate inside of the PCT cell. So that's something that you have to remember. So the enzyme, so if you block that enzyme, you're not only preventing the bicarbonate reabsorption, you're also promoting sodium, you're also preventing sodium reabsorption and promoting hydrogen excretion. So this will lower our body pH. That's why I told you guys that this drug leads to metabolic acidosis because it promotes bicarbonate excretion and also it promotes hydrogen excretion. So it will reduce the body pH. It will lower the body pH. There will be a metabolic acidosis. So that's something that you have to know. So by promoting bicarbonate excretion and hydrogen retention, so the hydrogen will be retained. Sorry, the hydrogen will be retained. Yeah, this exchanger won't be effective. So if you block this enzyme, if the carbonic and if the carbonic, if you block this enzyme, if the carbonic anhydrase enzyme is blocked, you will promote you will this exchanger will be inactive, and when this exchanger is inactive, it means there will be sodium in urine. The sodium will remain in the filtrate and the hydrogen will remain in the cell. So there will be hydrogen retention. Hydrogen will be retained in the cell, and as hydrogen is being retained in the cell, bicarbonate is being released, the bicarbonate is being excreted. So this will lower the body pH. There will be a metabolic acidosis. I hope you guys have understood I might have confused myself while explaining. I hope you guys have noted that. Please, if I have confused myself, please let me know. But right now, what the message that you have to understand is that these drugs, the carbonic anhydrase inhibitors, number one, they prevent bicarbonate reabsorption. Number two, they prevent sodium reabsorption. Number three, they promote hydrogen retention. So that's something that you have to know. And our long last is that it promotes metabolic acidosis. Yeah, so this forces sodium, water, and vast amount of bicarbonate to remain in urine. And by doing this, it also retains hydrogen. So that's something that you have to understand. So a continuation of our slide. So what are the indications? This drug, for those guys who have done ophthalmology, you have seen it being used, especially in the management of open angle glaucoma. The reason why it's very good is that it helps in decreasing the aqueous humor production. Yeah, so that's very good. It can be used in open angle glaucoma. So the acetazolamide is a very good drug when it comes to open angle glaucoma. Apart from that, also, it can be used for acute mountain sickness. Remember, for those guys who do mountain climbing, uh, the higher you go, there's a lower oxygen concentration. So one thing that you have to do is that we want the client to have a compensatory mechanism of hyperventilation. And the best way to do this is by giving the carbonic anhydrase inhibitor. Why? This drug will lead to metabolic acidosis. And when someone has got metabolic acidosis, the body compensates metabolic acidosis by hyperventilation. The body tries to get rid of CO2 in order to raise the body pH. So by doing that, the individual or the patient will hyperventilate, and we want that to be a compensatory mechanism when doing mountain climbing. It can also be used in correction of metabolic acidosis, of metabolic alkalosis. So in metabolic alkalosis, we have an elevation of bicarb ions, and when you have an elevation of bicarb ions, we want to remove bicarb ions. The best way to remove bicarb ions is through urine, and that can be done by administering carbonic anhydrase inhibitor. It will promote bicarb excretion, and that will correct the metabolic alkalosis. Some of the adverse effects, number one, we know it leads to metabolic acidosis because number one, as you excrete bicarbonate, you are lowering the body pH. And not only that, it also leads to hydrogen retention. So it propagates metabolic acidosis. Number two, it may also lead to hypokalemia. It may also lead to renal stones because there will be an alkaline urine. An alkaline urine promotes the formation of renal stones. So this is my diagram here that I've just generated from AI. So this is just an AI diagram that I have generated from AI. You can see how some of the indications when using the acetazolamide. Yeah, it's very good when it comes to open angle glaucoma. Yeah, but it helps in reducing the intraocular pressure. It's also good when treating altitude sickness because you want to induce the compensatory hyperventilation mechanism, and that will help when mountain climbing. Because remember, as the higher you go, in higher altitudes we have got very low oxygen level, and we want these people to be able to hyperventilate in order to breathe well. So that's a very good drug when it comes also to mountain climbing. So you've got other indications here, metabolic alkalosis. Obviously, when you want to, it can also be used for metabolic alkalosis when you want to get rid of bicarb ions. It can also be used in periodic paralysis, epilepsy, idiopathic, intracranial hypertension, and others. So this is some of the indications that I've mentioned. If I haven't mentioned any indication, you can put on the comment section. If I have mentioned something that is contradictory, please let us know, because this session is about helping each other and about understanding these drugs. So number two, let's move to the next diuretic. This is the osmotic diuretic. I want to stress this diuretic also is not so much used as an antihypertensive. It has other indications. Yeah. So the major drug here you guys have come across is the Mannitol. It always goes in as you can see, these are one of the formulations of Mannitol, intravenous formulation. As you can see, 20% Mannitol in 500 mils. This is how it looks like. Yeah, this the 250 mil, there's the 500 mil, and this you have got a 20% concentration. Yeah, so this is our Mannitol diagram. We know you guys. I'm very certain you guys have used it in your facility. And if you haven't used it, please, I will explain some of the indications whereby this fluid is very, very crucial. So mannitol, it may act on it acts on various sections. It may act in PCT, it may act on the descending limb of Henle, it may act on the collecting duct. So it has got a various size of actions, but I know it tends to work very well when it comes to working on the descending limb of Henle. So mannitol is a pharmacologically inert carbohydrate. So it's a pharmacologically inert carbohydrate that's freely filtered at the glomerulus but cannot be reabsorbed by the renal tubule. So once filtered, it cannot be reabsorbed. It remains in the filtrate. Yeah, it remains in the lumens. Yeah, so that's what it happens. And when it remains in the lumen, in the descending limb, remember in the descending limb, I told you guys, in the descending limb, water tends to move from the filtrate to the medulla. Now when you have mannitol, that won't happen, because the mannitol is an inert carbohydrate. It will lift the concentration of the filtrate higher than the medulla, retaining the water inside the distal tubule. Inside the descending limb. So a lot of water will be excreted. So they will not. So the osmotic pressure that tends to happen in the descending limb won't be there because of the presence of this inert carbohydrate. So that being said, there will be a lot of fluids in urine. So it remains trapped in the tubular fluid, creating a massive osmotic gradient that physically holds water within the lumen. Yeah. So the water will be held in the descending limb, because the descending limb will be highly concentrated than the medulla, and that is because of the presence of the mannitol, which we know it's an inert carbohydrate. So that's what you have to know. Then other thing that you have to know systematically, there's always a very adverse side effects when it comes to using mannitol. Why? Because before the mannitol reaches the kidney, it tends to increase blood plasma osmolarity. So there's always an increase in intravascular blood volume. So that's something I have to know. So how it draws intracellular water out of the swollen tissues and into the bloodstream. Yeah. So it tends to draw water into the intravascular space. So there's always a rise of the intravascular blood volume, which may have its own massive side effects. So some of the indications of using mannitol is: number one, reduction of elevated intracranial pressure. Is very good in managing cerebral edema, and also it may be used in elevation of the intraocular pressure, especially in the acute glaucoma. Apart from that, also it may be used in the prevention of the oliguric phase of acute kidney injury or the clearing of debris, like in the rhabdomyolysis. For those guys who have done medical surgical nursing, we know that rhabdomyolysis is the rapid breakdown of the skeletal muscles. And when you break down the skeletal muscles, we may have an elevation of the myoglobin, which is very toxic. We may have an elevation of the creatinine and other waste products that comes from the breaking down of the skeletal muscle. So that being said, we want to prevent the oliguric phase, yeah, and the best way is to using the mannitol. So some of the adverse effect, I've said before, that mannitol draws a lot of fluids into the intravascular space, and when that happens, there's always a sudden shift of fluid into the bloodstream that can rapidly trigger acute pulmonary edema or congestive heart failure. So mannitol may propagate pulmonary edema. So it's always contraindicated for those guys who have pulmonary edema. Don't use don't use mannitol to solve pulmonary edema. You'll be killing that client, yeah, because it makes it worse. It worsens the pulmonary edema because of the increased intravascular fluid volume. So there will be an increased blood volume, yeah, and this also happens in the capillaries that are found in the pulmonary tissue. So that's something that you have to note. So it may also lead to dehydration and hyponatremia, yes, because it drains a lot of fluid from the interstitial space, and that may lead to dehydration. So these are some of the indications that I've derived from an AI image. You can see it can be used in cerebral edema. I think this is the major. These two, the major indications of mannitol is, I think, is cerebral edema and the increased ICP. So these two are the major indications when it comes to using mannitol. It can also be used in acute glaucoma, as you can see here. It can also be used as renal protection. especially in rhabdomyolysis. Rhabdomyolysis may lead to acute kidney injury because when there's a rapid breakdown of skeletal muscles, there'll be an increase in myoglobin, and myoglobin is very much nephrotoxic. So to prevent kidney damage, especially in the oliguric phase, we may use the mannitol. It's also used to maintain diuresis, especially in the oliguric state. I've also mentioned this one. So these are some of the summary when it comes to the indications of using the mannitol. So I believe you're together. So if you haven't understood what I've mentioned, you can always forward backwards to recap what I've just said. So let's move to the loop diuretics, number 3. So I think this is the most used diuretics. Yeah, it's very much common in our facility, very common when it comes to examination. So for those guys who are sleeping, or if you are not alert, please be alert for this section because I want to dig deep on how we use the loop diuretics. So number one, loop diuretics are the most potent class because they target a segment responsible for reabsorbing roughly 25% of filtered sodium. So where does loop diuretics work? It works on the thick ascending limb. I believe you guys remember. So some of the drugs that are used here, the furosemide or the Lasix. Yeah, we have the bumetanide, we have the tosamide, we have the ethacrinic acid. The ethacrinic acid, these are non-sulfonamide alternative because remember these drugs also, they contain sulfur. So that's something that you have to note when it comes to using these drugs. But the most commonly used drug here is the furosemide. So this is the Lasix. This is the IV formulation. I believe we also have an oral formulation. We also have the tosamide injection. We also have this is the bumetanide injection. Yeah. So despite of IV, we also have got the oral formulations of these drugs. That's something that you have to note. So this drug is very common. You guys have used it in your facility. So such a function here is the thick ascending limb. Remember, I told you guys how it works. It targets this co-transporter, the sodium calcium chloride co-transporter, and I explained how this co-transporter works. If you haven't understood, forward backwards and recapture on what I explained when it comes to this co-transporter. I explained the physiology that happens in the thick ascending limb. So if you haven't if you didn't understand, you can always forward backwards and see what I was trying to explain. So this drug, they inhibit this co-transporter, the sodium potassium chloride transporter. So by blocking this transporter, they prevent the reabsorption of sodium, potassium, chloride, and also calcium and magnesium, as I explained. Yes, they also lead to they also decrease the reabsorption of calcium and magnesium. So the indications when it comes to loop diuretics is very good when it comes to managing acute pulmonary edema. Yeah, it can also be used in chronic congestive heart failure or excavations. It's very good also for managing hepatic cirrhosis with ascites. It can also be used in nephrotic syndrome and advanced chronic kidney disease, and also hyperkalemia because it helps in getting rid of potassium ions. Yes, it can also be a secondary line in hypertension. Why? Because remember, the loop diuretics, they really help in getting rid of fluids. And when you get rid of fluids, you are decreasing the intravascular fluid volume. And if you decrease intravascular blood volume, you are decreasing the venous return, you are decreasing the preload. If you decrease the preload, you are decreasing the stroke volume. If you decrease the stroke volume, you're decreasing the cardiac output. If you decrease the cardiac output, you're decreasing the blood pressure. So that's why these drugs can be used as second-line agents when it comes to managing hypertension. So adverse effects, number one, they may lead to hypokalemic. They help in getting rid of potassium ions, and this may lead to hypokalemic. Yes, it may also leads to metabolic alkalosis. Hypomagnesemia, hypocalcemia, I've explained how that happens. Yes. Remember, these drugs also lead to ototoxicity. So when these drugs are given through intravenous doses, be very cautious, especially when used in combination with the aminoglycosides. These two combo may lead to severe ototoxicity. So they also lead to they may damage the hearing function. So that's something that you have to note. Yes, they may also lead to hyperuricemia. Yes, they compete for the organic acid secretory transporter in the proximal tubule. So these drugs may also lead to, so they prevent they prevent the excretion of uric acids. Yeah, they promote the retention of uric acid, and this may lead to hyperuricemia. So that's something that you have to note when it comes to using these drugs. It may also lead to dehydration, obviously, and acute kidney injury, which obviously dehydration leads to prerenal prerenal AKI. So these are drugs that I extracted via AI, just to help us understand some of the indications that comes when using the loop diuretics. So remember, first you know that some of the drugs that fall under this category are the Lasix or the furosemide. You have the bumetanide, you have the tosamide, you have the ethacranic acid, and others. So it's very good when it comes to edema. Very good. I think loop diuretics are the best agents when it comes to managing edema. So they're very good in edema. They also, yes, I've explained edema obviously in liver cirrhosis with ascites, pulmonary edema. Yes, I've explained that one. They may also be used as second line in hypertension. Yes, they may also be used in hypercalcemia. Why? Because they promote calcium excretion. So if someone has got a elevation of calcium ion in serum, we may want to promote calcium excretion. And the best drug that does this is the loop diuretics. They promote calcium release in urine. So they may also be used in hyperkalemia. Yes, we may also want to reduce the amount of potassium in blood. And the best way to reduce potassium in blood is by promoting potassium excretion in the kidneys. And the best drug that does that is the diuretics, the loop diuretics. So that is also a good drug to be used. And also they can be used when it comes to those guys who have drug or toxin overdose. We may want to promote diuresis, and the best drug that promotes diuresis is the loop diuretics. So this is overall indications when it comes to using the loop diuretics. Now, as we move further forward, I want us to look on the thiazide diuretics. Some of the drugs, so the thiazide are moderately potent agents that act on early DCT, where we have 5% of filtered sodium, where 5% of filtered sodium is always reabsorbed. So this is where, so in the thiazide diuretics act on the early DCT. The early DCT, we know in the early DCT, we always have around 5% of filtered sodium being reabsorbed. And the drugs that fall under this category are the hydrochlorothiazide. So the thiazide is the suffix that you have to see in this drug, the thiazide. So the hydrochlorothiazide, the chlorothiazide, we also have the clothalidone and the metolazone. So that's our drug, our formulation of the chlorothiazide. Yes, this drug, this losartan potassium. This losartan potassium. This is one of our angiotensin receptor blocker. It's always used in combination with hydrochlorothiazide when it comes to managing hypertension. They are a very good drug, as you can see. I'm very certain you guys. I'm very sure you guys have come across this drug, especially those guys who are in medical wards, dealing with those clients who are hypertensive. I've seen this drug is very much famous when it comes to managing hypertension. We also have the hydrochlorothiazide tablets, as you can see, 12.5 milligram. We have our chlorothiazide injection. Yeah, so these are some of the drugs that are being used. There are others. There are very many. If you know of a drug that you have used in your wards or have come across, please post on a comment section for us to understand and learn better. So I've said that they act in the early DCT. I won't repeat it. I explained how this happens in our previous slides. So they inhibit the apical sodium chloride cotransporter. I have explained how this transporter works. This transporter, it takes sodium and chloride from the filtrate to the cell. Yeah, that's how it happens. The apical sodium chloride cotransporter. So unlike loop diuretics, thiazide they always increase calcium reabsorption. Explained in my previous slide how this happens. So if you haven't watched, please forward backwards, watch on how I have explained what happens in the early distal convoluted tubule, how the calcium retention happens, especially when it comes to using these drugs. So one thing that you have to note that thiazide diuretics, they lead to calcium reabsorption. So if a patient has got hypercalcemia, if there's a elevation of calcium in the blood serum, you have to stop using these drugs, yeah. Or if a patient has got hypocalcemia, yeah, this drug is very good because it leads to calcium retention. So that's something that you have to note. This when using these drugs, they increase calcium reabsorption or retention. So the reduction in intracellular sodium enhances the basolateral sodium calcium exchange activity, pulling more calcium out of the tubular fluid into the interstitium. I've explained. So these drugs, what happens? These drugs, they tend to inhibit this co-transporter, and when you inhibit this co-transporter, there will be low sodium in the cell. There will be low sodium in the early DCT cell. So the low sodium in the cell will propagate, will activate this exchanger. How? This exchanger will bring sodium in the cell and take more calcium to the blood. So that's why we always have calcium retention when using these drugs. So what are the indications of using thiazide diuretics? So they're very good when it comes to treating primary essential hypertension. They're very good also in dealing with nephrolithiasis. Remember, in nephrolithiasis, we have got calcium in urine. And if you want to decrease calcium in urine, you have to increase calcium reabsorption. So the best drug that does this are the thiazide diuretics. They always promote calcium retention. So when there's calcium retention, there will be a decreased calcium in urine, and that will be beneficial when it comes to nephrolithiasis. Nephrolithiasis, basically, these are the renal stones. Then they are also good for mild to moderate edema in heart failure. They can also be used in osteoporosis, because in osteoporosis we want to increase the amount of calcium in blood, and the best way of increasing calcium in blood is by using the thiazide diuretic. So they're very beneficial in osteoporosis. Yeah, they're also good when it comes to dealing with nephrogenic diabetes insipidus. How? So what it does, that it reduces polyuria by inducing mild volume depletion, which increases proximal tubule water reabsorption. So this drug is very, very important when it comes to managing nephrogenic diabetes insipidus. Yeah. Yeah. So what happens that when using this drug, it reduces polyuria by inducing mild volume depletion, which increases proximal tubule water reabsorption. So by using this drug will promote water reabsorption in the proximal convoluted tubule. So that's something that you have to understand. Then adverse effects number one is hyponatremia. Yes, it leads to sodium loss than water. Then it leads to hypercalcemia. There will be an increased calcium retention. So if a client has got increased calcium, these drugs should be stopped. Yeah, because they will make it worse. Apart from that, they may lead to hypokalemia and hypomagnesemia, because they also allow more proton in urine. They tend to allow more potassium in urine. They can also be used in metabolic alkalosis. How? Because it allows more proton in urine. So they promote the release. They allow more. They promote the release of hydrogen ions in urine. So by promoting hydrogen loss, they will increase the pH, promoting metabolic alkalosis. Just like thiazide diuretics, they also block the excretion of uric acid, leading to hyperuricemia. So that's something that I have to note. Another unique thing about thiazide diuretics, that they also lead to hyperglycemia. They are known to inhibit insulin production and sensitivity, so they may also lead to hyperglycemia. So for a diabetic client using these drugs, be very vigilant, because these drugs may propagate hyperglycemia. They may also lead to hyperlipidemia. So this is an AI image that I've derived on the indications of using thiazide diuretics. It can be used in managing primary essential hypertension. It can be used in edema, especially those that are due to congestive heart failure. It can also be used in edema due to liver cirrhosis, like the ascites. It can also be used in CKD, edema caused due to CKD. It can be used for those clients who have got calcium renal stones prevention. It can also be used in nephrogenic DI and also be used in hypercalciuria. So this is some of the indications also. And lastly, they may be also be added in patients with persistent edema despite loop diuretic therapy. So it can be used together with loop diuretics for those guys who have got persistent edema despite of using the loop diuretic therapy. So the key point here that does it at the best for long-term control for hypertension and mild edema, and for specific benefits like reducing urinary calcium. So they are also known to reduce calcium in urine. Now let's move to our last type of diuretic. That's the potassium sparing diuretics. So potassium sparing diuretics, I think there are two mechanisms of action that act. Now, so these agents have a weak diuretic efficacy when used alone. Why? Because they target less than 3% of filtered sodium, but they are clinically essential for preserving potassium levels. That's why you have got potassium sparing. These are diuretics that help in sparing potassium. So they fall into two subclasses based on their molecular approach. So we have got two mechanisms of action when it comes to potassium sparing diuretics. Now, the first one, we have got aldosterone receptor antagonist. So these are the drugs that prevent the production or the attachment of the aldosterone. So these drugs, they literally stop the function of the aldosterone hormone. And remember, I told you guys that aldosterone hormone promotes the manufacture of three channels, of the NAC channels, of the potassium channels, and of the sodium potassium ATPase pump. So if you prevent the attachment of the aldosterone hormone on its receptor, it means the three channels won't be formed. And that being said, there will be sodium excretion in urine, there will be potassium retention. So the two drugs that work as aldosterone receptor antagonist are the two drugs called spironolactone and eplerenone. Guys, for those guys who have watched our third episode when we discussed RAS, the renin-angiotensin aldosterone system, I mentioned these two drugs. So for better understanding, you can watch that third episode. It will help you understand better on what I'm trying to elaborate in this slide. So these two drugs, the major action is that they are aldosterone receptor antagonist. They literally prevent aldosterone from conducting its function. So they act on the late. So the set of action here is either the late distal convoluted tubule or the collecting duct. So those are the two key areas under which these drugs act on. So the mechanism of action, I've said that they're competitive antagonists that bind to the intracellular mineralocorticoid receptor. It's a very hard term to pronounce. But this means in short, they compete with aldosterone. Yeah, so they antagonize aldosterone. So this blocks the nuclear translocation of the receptor, preventing the transcription of genes that synthesize epithelial sodium channels, the NAC, the sodium potassium ATPase, and also this potassium channels. The second classification in terms of the mode of action, we have got the direct NAC blockers. So these ones, they block the NAC channels, they block the sodium channel. And the two drugs here are the amiloride and the triamterene. Amiloride and the triamterene. Yeah, so they act on the collecting tubule. So the mechanism of action here is that they block NAC channels on the apical membrane of principal cells, independent of aldosterone. So these drugs, they block the NAC channels, the sodium channels, and when they block the sodium channels, they will inhibit sodium reabsorption. If you inhibit sodium reabsorption, it means that the sodium potassium ATPase pump will be inactivated, yeah, and that will lead to the retention of potassium. So this prevents sodium entry, reducing the negative lumen potential and eliminating the electric driving force that normally secretes potassium and hydrogen into urine. So it means there will be retention of potassium and hydrogen in the blood. So you have two drugs. So you've got the two drugs. So these two, the spironolactone and the eplerenone. So the spironolactone and the eplerenone, these two drugs are the aldosterone antagonist. And the other two drugs, the amiloride and the triamterene, these two drugs are the ENaC channel blockers. They block the sodium channel. So what are some of the indications of using these drugs? Number one, they can be used in counteracting diuretic-induced hypokalemia. That's why they're always co-administered with loop or thiazide diuretics, because if there's hypokalemia and you want to use an antihypertensive, the best one is the potassium-sparing diuretics because they help in the retention of the potassium. So they're also good in primary and secondary hyperaldosteronism, especially the spironolactone and the eplerenone. The aldosterone antagonists are very good when it comes to treating hyperaldosteronism because in this condition, there's elevation of aldosterone hormone. And when there's an elevation of aldosterone hormone, you want to block their work. You want to block. The aim is to block their action, and the best drug that do that are the drugs that antagonize aldosterone. They can also be used in heart failure with reduced ejection fraction to improve survival and reduce cardiac remodeling. So these drugs are also good. They're known to reduce mortality. They're very good drugs. It can also be used in hepatic cirrhosis with ascites as first line, especially the spironolactone. Adverse effect number one, hyperkalemia. Obviously, these drugs retain a lot of. They tend to retain a lot of potassium. So if you have this, if the client's using this drug, you have to be very alert on the signs of hyperkalemia. If you don't know the signs of hyperkalemia, watch our third episode. I explained the signs of hyperkalemia and how you can notice, especially on EKG, hyperkalemia. There are ways in which we can easily identify hyperkalemia on EKG. If you see on EKG we have got a peaked tented T waves or a wide QRS, then you can easily denote that this client likely has hyperkalemia. It also leads to metabolic acidosis because remember they lead to the retention of potassium and sodium and hydrogen ions. So the retention of hydrogen ions will lead to metabolic acidosis. So another adverse effect, especially when using the aldosterone antagonist, is that they not only block aldosterone hormone, they also block other special hormones. Yeah, and this being said, it may lead to gynecomastia, erectile dysfunction, and menstrual irregularities due to non-specific binding to progesterone and androgen receptors. So the drugs that block aldosterone hormone, they may also block androgen, and when they do that, they may lead to gynecomastia. Gynecomastia basically is breast in males. If you see a male client having breasts, it may be gynecomastia or man boobs. A drug that tends to cause man boobs are the spironolactone. Eplerenone is highly selective and carries much lower incidence of these side effects. So these are some of the indications that have derived from an AI. Some of the indications. Number one, hyperaldosteronism. Yes, you want to use the aldosterone blockers, the likes of spironolactone and the eplerenone. Yeah, they're very good drugs when it comes to dealing with hyperaldosteronism. Number two, they're also good when it comes to dealing with heart failure with reduced ejection fraction, especially the spironolactone. So research has shown that this drug improves survival rate for those clients who have got heart failure with reduced ejection fraction. It can also be used for cirrhosis with ascites. It can also be used to prevent and treat hypokalemia because it leads to potassium retention. It's a very good drug when it comes to dealing with resistant hypertension. I've explained this one, I think so. It can be used for edema, it can be used in nephrotic syndrome, it can be used for PCOS, polycystic ovarian syndrome. and others. So these are some of the key points. Our key point here down here is potassium sparing diuretics are indicated to counteract aldosterone excess, manage heart failure and cirrhosis, prevent hypokalemia, and provide benefits in selected conditions while conserving potassium. So this is a very key note that you have to remember. That's that the main indications of using potassium. Number one is the potassium sparing diuretics. They really help in counteracting aldosterone excess. Number one. Number two, managing heart failure and cirrhosis. Number three, preventing hypokalemia. Number four, provide benefit in selected conditions while conserving potassium. They lead to potassium retention. Wow, that's all about our nice session. It was amazing. I'm exhausted, but I think it's around 2 hours, around 1.5 hours. It was very, very, very long. But at the end of this section, we have not only looked on the diuretics as antihypertensive, we have also looked on diuretics as a topic on its own. So it was amazing having this session with you guys. Thank you for your patience. Thank you for being for supporting our channel. I believe we will have our last episode on antihypertensive series, and we will be able to understand all these drugs that are used in managing hypertension. Thank you for your patience. If you are new to our channel, please click the subscribe button, like, comment, click the notification button also, so that when you have got a new episode, you are notified. And also share our link on various platforms, be it our Facebook account, be it our Twitter account, be it our TikTok account, be it our IG account. Our aim is to expand and to have more viewers. So thank you for this section. If you have any new message, please post on the comment section. If you feel that there's something that has been mentioned that is contradictory, please let us know. And I appreciate you guys for your patience. Thank you, and may God bless you all. Goodbye.